Good afternoon, everyone. I want to thank you for coming today and welcome you to our next Congressional Biomedical Research Caucus briefing with Dr. Atul Butte. I first of all want to thank the Congressional Biomedical Research Caucus co-chairs. That would be Congressman Rush Holt, Congressman Michael Castle, and Congressman Brian Bilbray. They've been great supporters of our caucuses, and we're very much indebted to them. I also want to remind everyone that our next caucus would be next Wednesday in the same room at 12 o'clock, as usual, with lunches as well. And also, if you can't ever make our caucuses or you would like to see past caucuses, they stream live on the Coalition for Life Sciences website, and that's www.coalitionforlifesciences.org, as well as podcasts are available. You can sign up for the RSS feed. Now, on to today's caucus. I would like to introduce Dr. Atul Butte. He is an assistant professor in medicine and pediatrics and by courtesy computer science at Stanford University and the Lucile Packard Children's Hospital, and is a pediatric endocrinologist. His laboratory focuses on solving problems relevant to genomic medicine by developing new methodologies in translational bioinformatics. Dr. Butte has authored more than 50 publications in bioinformatics, medical informatics, and molecular diabetes, and has delivered more than 70 presentations worldwide on bioinformatics. His research has been featured in the 2008 New York Times Science Times and the International Herald Tribune. And if you didn't pick up a copy of the New York Times piece, we did have it outside for you, and we can make sure you pick one up on your way out. Dr. Butte's research is supported by grants from the Howard Hughes Medical Institute, as well as several from the National Institutes of Health, the California Institute for Regenerative Medicine, and also PhRMA, which is Pharmaceutical Research and Manufacturers of American Foundation. So without further ado, please join me in welcoming Dr. Butte. Thank you.
Thank you. Thank you very much. Thanks for that introduction. So if there's one term that explains what it is that I do in the lab, I think the term that really I've got stuck on is this term called translational bioinformatics. So I'm going to spend a slide explaining what that actually means. So maybe, especially based on some of the previous speakers that I saw, you've heard this term bioinformatics, which is the application of computers, computer science to biology and medicine. I think bioinformatics has now been around for 30 plus years. We know that there is application. We know there's a lot of data being collected in life science. I'll explain a lot of that. But I think the new part is this translational term. And so we also have this term that's floating around medical schools and medical centers called translational medicine or translational biology, where we certainly know we've collected a lot of data because of the genome era. We have a lot of clinical data, proteomic data, genomic data. But where are the tools for patients and healthcare providers? That's what it comes down to. And so we have this new mission of translational medicine or translational biology, which is essentially to take the discoveries from the bench and get them to the bedside. That's what we've now called T1 translation. It's actually a T2 translation, which is to take it from the bedside and actually get it to the community. In other words, out of the academic centers and into the general population as well. So we've got bench to bedside to community. That's what translational medicine means. So now we've taken these two terms, translational medicine and bioinformatics, and really hybridized them both together. Because I think bioinformatics certainly has led to this problem. There is a well-known problem of taking these discoveries from the bench and getting to the bedside. We wouldn't have this term. We wouldn't have these missions if we didn't have a problem here. But I think bioinformatics is in a great position to solve this problem. So I'm going to show you a couple of clear-cut examples how bioinformatics enables us to actually come up with novel diagnostics, novel therapeutics. And really the punchline is actually we can do this using publicly available data. Now, we get a lot of inspiration from this quote from Elias Zerhouni, who is the outgoing director of NIH. Elias Zerhouni, in this paper in the New England Journal of Medicine in 2005, wrote that at no other time has the need for a robust, bidirectional information flow between basic and translational scientists been so necessary. It has been particularly pleasing to me and others that we had a director of NIH that used words like robust, bidirectional information flow. You can imagine previous directors of NIH did not use such terms like information flow. Elias Zerhouni himself was an imaging informatician. He was working on cardiac MRI images, trying to figure out function of the heart from that. And so this has been good times for bioinformatics. And bioinformatics has really gotten to the top tier in terms of when you even look at the stimulus projects and the ARRA projects, bioinformatics, biomedical informatics, it actually has a top line in the categorization. I mean, it's established that we need to develop more in bioinformatics. So translational bioinformatics is the development of analytical, storage, and interpretive methods that optimize taking all this voluminous data and coming up with tools for patients, providers. That's the kind of definition here. I'm going to spend most of my time talking about these gene chips, or microarrays. Now, David Botstein spoke earlier in the series, so some of you who've heard that can tune out for a slide or two. But this is now a 13-year-old technology, and it still seems pretty nifty that we have this technology. Essentially, RNA expression microarrays are measuring RNA, or the blueprints on their way to coding for proteins. You know, we have the DNA in the middle of the cells. The cell decides to make protein. For example, your cells are making protein because you're chomping on some protein in the turkey, or you're eating some fat in the mayo. You're making some proteins right now. And the way that the cells do it is to make the RNA, the blueprints on their way to coding for protein. It turns out it's very easy to measure RNA, because most of the RNA looks the same no matter what it's coding for. It can be an acidic protein or basic protein. It doesn't matter. The RNA essentially looks the same. The difference is in the letters, the spelling. So now we have these gene chips, or microarrays, that are the size of your thumbnail that can quantitate, measure, amount of every gene in the genome. Okay? That's already a 13-year-old technology. Today, they're commodity items. Commodity means we don't even really truly care which company we're buying them from. Which is the cheapest provider, is usually the first question we ask now. So they're commodities. Whole genome, single chip, and they go for about $200 to $300. Okay? Now, because of that, everyone started to run microarrays over the past 10 years at every major institution, even the minor ones. Because of that, all the journal editors said, no more papers on microarrays, unless you put your data out into an international repository. Okay? And the precedent for that had been set 26 years ago by GenBank. GenBank got its start because even after the first paper where the DNA sequence came out, people realized they really got tired of typing in these sequences. A, G, T, C, let's not make a mistake. So that's pre-web, that's even really pre-email in some ways. People were shuffling tapes around, hard drives around. You get the picture. The idea was to share data. So this is essentially the technology I'll spend most of the time talking about. This is what one of them looks like. In biology and medicine, we like 96 versions of things. So this is a 96 well plate version of this. One of these in the middle of each of these. Now, Todd, so the seminal work in this field came out about 10 years ago, just showing, for example, that gene chips measure every gene in the genome. We can use this to figure out things that are hard for doctors. Here's an example. Here's a picture of acute lymphocytic leukemia. Picture of acute myeloid leukemia, ALL, AML. Believe me, as a pediatrician, I can tell you it makes a world of a difference on day one which one you're telling the parent that their child has. If their child has ALL, the discussion usually revolves around you telling them to plan for a discharge from the hospital in a month. When it's AML, the discussion usually revolves around whose bone marrow can we transplant into your child, okay? World of a difference. And you would want to know that on day one. It's not just this projector. You really have a hard time telling these two apart. And what Todd Golub showed 10 years ago is that you can use these gene chips and just measure every gene in the genome and find genes that are high in ALL, low in AML, or vice versa. No matter how you do this, you end up with 200 or so genes, any one of which can now be used as a diagnostic test to help us tell, is this ALL or AML? Another seminal work came from Ashley Lizadeh and others at Stanford and National Cancer Institute, looking at it from a different perspective. These are folks using these same microarrays, looking at one disease, not two diseases, one disease, B-cell lymphoma. And they took a bunch of samples of B-cell lymphoma, put them on these gene chips, measuring every gene in the genome here, and lo and behold, they found out that there was actually a 50-50 split. Either you had a pattern like this group of individuals or a pattern like this group. They were high in these genes, low in these, and vice versa down here. You were either in this group or that group. Well, actually, as physicians, remember, we couldn't tell these two groups apart. They all looked like they had the same diagnosis for us. They were all treated the same way. But when you actually look at them in a molecular perspective, all of a sudden we realize if you belong to this light blue group, those patients actually had a much harder time and actually died sooner with this disease than the other half. So all of a sudden, the molecular measurements now are telling us a clinically important difference between one subtype and the other subtype that no physician was able to tell prior to that. Indeed, in some institutions like the Netherlands Cancer Institute, this is routine care now. It's not even considered research measurements. These are routine care now to make these kinds of measurements. Where are we going with this? On the genetic side, certainly you've seen the lay press and other sources where these same microarrays now can measure SNPs, or the differences between our genomes. We estimate that there are about 10 million differences between any two individuals in terms of the DNA, 10 million. Current chips, again, the same size of your thumbnail, can measure 2 million of those differences today for about the same commodity price, $300. We can look at 2 million of these differences. That's what's currently available. Just out over the past year are high-throughput sequencing measurements, where now for a cost of about anywhere from $30,000 to $100,000, instead of just looking at the 2 million differences, just sequence the whole genome. So we have multiple companies that offer those kinds of products. And then we have the Jiffy Lube of the genome. This is actually Discover Magazine's title for this, where we're expecting, so this particular company announced last summer that by 2010, and they already have a science paper out on this, by 2010 we should be able to sequence the entire human genome in 15 minutes at the cost of a couple $100. So about the cost of an oil change, faster than an oil change. Actually, 25 times coverage of your own genome here. But what's been the most disruptive thing, actually, in terms of research for me, okay? This is cool. This has been going on for a decade now. What's been the most disruptive thing for me is the public availability of this raw data, okay? So I told you a story about the journal editor saying no more papers on microarrays. They wanted international repositories to hold this data. So one of the major repositories for this kind of data, in fact, most of them are all like GenBank. They're all run out of the National Library of Medicine, which is right down the road on the NIH campus. So if you go to the National Institutes of Health and the National Center for Biotechnology Information, run out of the National Library of Medicine, the repository for those gene chips, those microarrays, is called the Gene Expression Omnibus. Now, one of the most striking things is this number here. It's a couple of weeks old now. There are already over 300,000 of those chip measurements publicly available. Okay? 300,000. This number doubles or triples each year. That's a growth rate faster than Moore's Law, where in Silicon Valley we know Moore's Law well. That's computational power doubles every 18 months. Here we're talking about doubling every 12 months. Okay? Exponential, but faster. So let me paint a kind of interesting picture to you. Here's a search box here. A 12th grader today who now needs to do a science fair project can now type in breast cancer and actually be able to download 20,000 samples of breast cancer today. That's more samples than a breast cancer researcher has today, right? Because a high school student can get to all of any one investigator's data plus all of their competitors. Indeed, 2 of the 300 Intel Science Talent Scout semifinalists last year came from my lab, enabled in this way. 2 out of the 300 came from my lab. You type and you get more samples than any investigator in any field. And if you're not happy with the number, next year it will be double the number at any point. Okay? It's an amazing world. That high school student can download 20,000 samples of breast cancer just as easily as she can search for a song on iTunes. Okay? It's not just the RNA blueprint side. This is on the DNA side as well. dbGaP, again run by the National Library of Medicine. Multiple studies on Parkinson's, schizophrenia, lung cancer and smoking, addiction, ADHD. And you can see 1,900, 2,000, 4,000, 5,000. This is on the DNA side. Now, of course, here, because of privacy, you can't just click to download the data. You click to download the forms to fill out to get permission to get to the data. It's a slight subtlety, but you get the idea. Public availability of data can enable new kinds of research to be done. So I'm going to step you through one such project that we had in the lab. This is what was described in Science Times last year. I'll walk you through this. My hero of the last few years now has been Linnaeus. Those of you who might have majored in biology might remember Linnaeus. You all had to remember at one point in elementary school, kingdom, phylum, class, order, family, genus, species. You all had mnemonics for that. Some were clean, some were dirty. You don't have to repeat them here. But every one of you had to know that in elementary school. That's because of Linnaeus. In fact, Linnaeus, we just celebrated his 300th anniversary of his birth, just like we're celebrating evolution. I think we celebrated Linnaeus 2 years ago. And everyone remembers him for that taxonomy of species. One thing that we completely forgot about Linnaeus, even at all the Linnaeus parties 2 years ago, was actually Linnaeus was the first guy to put diseases into a taxonomical tree. There's a word for that called nosology. The word itself came in the 1700s, a systematic classification of diseases. And Linnaeus published this book in 1763, which was the kingdom, phylum, class for diseases, not for species, for diseases. Why did everyone forget about this? Because Linnaeus got it horribly wrong. As you can imagine, nobody was doing autopsies at the time. Microscopes and things were just still not there. There's only so much you can tell about a human body or why someone died using the tools of the trade. But we do use nosologies today. In fact, the nosology that we use today, the most common is the ICD system, the International Classification of Diseases. I'll step you through the history of that in a moment. The pathologists use one called SNOMED, but even SNOMED, so ICD started in the 1860s, a little after the American Civil War, basically. We use the same system it's really evolved since the 1860s. The SNOMED system, the pathologists, they can look under a microscope, they do a little bit better, but even that's about 78, almost 80 years old as well. Our thinking is, why can't we build a brand new taxonomy of all of human disease? Because we have so many people making these measurements and contributing them into the repository. What would happen if we built a brand new taxonomy just using the public data? So just to step you through nosologies and ICD, I want to make a point about them. This is what the ICD system looked like 100 years ago. Exactly 1909, this is the Manual of the International List of Causes of Death. This wasn't a WHO standard at the time, of course, because this predates the United Nations. This was a League of Nations standard, it turns out. Okay? And you can see it's listed the same way we do our codes today. If you've ever spent any time in a doctor's office, you know all the check marks at the back of the sheet that they give you to take back to the receptionist. And it's the same way back in 100 years ago. Code 39 is cancer of the buccal cavity, the mouth, cancer of the stomach, liver, intestines, female genital organs, breast, skin. And then they do what we do all the time in the ICD system. They said, Code 45, boy, we're running out of codes. No more codes for cancer. All the other cancers should be in Code 45, including lung cancer and brain cancer. In other words, it's not even worth distinguishing lung cancer from brain cancer. They should both get the same code. The number one cancer killer, I think now, is lung cancer, was just cancer not otherwise specified 100 years ago. Essentially, that's a smoking issue. Code 50 was diabetes. It doesn't matter if it's juvenile, adult, type 1, type 2, because it would still be 10 years later that insulin was discovered. In fact, my entire field of endocrinology did not exist. They were all just listed under general disease, except for one, the thyroid gland. For some reason, I guess they thought it affects how you breathe, so they put it under the respiratory system. Smallpox, cholera, plague, glanders, and anthrax. If I had a single patient of any one of these in our institution, I'm sure Homeland Security will be visiting us the next day. They're all bioterroristic agents. They had the lowest codes in the book. They were just prevalent at the time. And this international standard literally has to say, if you see a death certificate where a patient dies because of a visitation from God, be sure to code it as 189. So here's the point. The same way we're chuckling at this, imagine how they're going to be chuckling at us even 20 years from now. Those silly fools didn't know this arthritis and this bowel disease were the same thing, right? That's the point of having a brand new taxonomy from the molecular side, not just how the doctors take care of it, for the molecular side. So we build tables like this with hundreds, if not thousands, of diseases going this way and tens of thousands of genes this way, and all from the public repository. We know how much each gene changes in each disease. We can build this. We have tools to automatically read the descriptions, and we can make a stab at this. And in the end, here's what a new tree of diseases looks like. I'm not going to walk you through all of this. But you get things like colon cancer and colon polyps are next to each other. That's good because we biopsy those polyps when they do colonoscopies. But what got us written up in the New York Times was a tiny branch over here. The heart attack, myocardial infarction, is right next to muscular dystrophy. Everyone knows what a heart attack is. Muscular dystrophy is this congenital muscle weakness. Kids are born with this. Eventually they can die because of their muscle failure, the heart muscle failure, but the telethon. Why is this interesting? Because today we have 40 FDA-approved drugs to treat heart attacks, but we really don't have any to treat muscular dystrophy. And I'm telling you, from the molecular perspective, the process of going from normal to heart attack is pretty similar as going from normal to muscular dystrophy. So why can't we try one of those 40 drugs in a kid with muscular, even before you put in a kid? Why can't we try one of those 40 drugs and in a mouse model of muscular, or even a cell line in a dish? The idea is we have these drugs that work for certain diseases. Why can't we share them to other diseases? So the idea is called drug repositioning or drug repurposing. There are some classic examples of this. The first such example is a drug everyone's heard of. It was initially tried for a heart condition, and all of a sudden they had an annoying side effect of actually causing hair growth, which is now minoxidil. So now we use it as a gel for hair growth. But of course, the classic example, of course, is Viagra, which started as a cardiac trial, annoying, interesting side effect, no one returning the drugs, and you get a new indication. Now, in fact, Viagra has done this again. So Viagra has gone and gotten another indication for pulmonary hypertension. When the blood vessels that go from the heart to the lungs get constricted, it turns out Viagra can help with that as well. So it's gotten 2 new indications in that way. But those are usually found by side effect and by accident. Maybe we can predict new uses in this kind of way. So the other way we can use this kind of new thinking of disease is to actually come up with real new diagnostics for patients. I'm going to walk you through one example of that. There are millions of, hundreds of thousands, if not a million transplants done in the United States and worldwide. Transplants meaning you need a new kidney, a new heart, a new lung, new intestines. And one of the major problems with transplants is actually the process called acute rejection. All of a sudden, the body wakes up that morning and says, this wasn't part of me anymore. I need to get rid of this. Acute rejection. The only way to really diagnose acute rejection today is to stick a long needle into the thing that you transplanted in, hope you hit something that looks like rejection, and look under the microscope. That's not a particularly convenient way to diagnose it, especially since there are no symptoms. There's no pain. You don't feel pain when you're getting rejection. It's a cellular thing. So we wanted to find a new blood test for rejection. But you know what? We want a universal blood test for rejection. We don't care if a heart went in or a lung went in or a kidney went in. We should be able to find one blood test for rejection no matter what organ went in. So how does a bioinformatician do this? Well, publicly available data. We start with publicly available data of kidney transplant with rejection, adult kidney transplant and pediatric kidney transplant with rejection, and even a heart transplant with rejection, all publicly available. This one was particularly old, so we talked to the investigator. We got a fresh data set there. These two are straight from the public repository. And we looked to see what's in common. 45 genes are in common. It doesn't matter if it's a kid or an adult or a heart, 45 genes all went up in rejection. So we test all 45. Well, actually, 10 of them were available in kit form. Biologists tend to be lazy sometimes. Instead of building our own tests, let's just go buy the kit ones first. 10 out of the 45, we said, let's just pull the trigger on all 10 of these, $500 each, to be honest with you. Okay? Test all 10 in new serum samples to see, can we actually do any of these become new blood tests? Out of the 10, 3 of the 10 are brand new blood tests for rejection, validated in kidney transplant. So here's low and stable, up in rejection, up in rejection, up in rejection. Very good sensitivity and specificity. In fact, when you look under the microscope for one of them, it's rip-roaringly high in the kidney, as we would have tested, but also in liver and heart. So high in heart, liver, and kidney rejection. Doesn't matter what organ it was that went in, but these tests are high. This was enough to convince our collaborators to thaw out their precious heart transplant serum samples, and all 3 are up in heart transplant as well. So here's an example of how we have not just 1, but 3 brand new blood tests for acute rejection. This is pilot data. Okay, we need to do clinical trials on this. 3 brand new blood tests for acute rejection, regardless of the organ, that we found with the help of publicly available data. Okay, the same data any high school kid can get to. So my take-home points, the public availability of research data truly enables novel therapeutics and diagnostics to be discovered. Why am I making such a big deal of this? Because there are scare words now, like HIPAA. Okay? HIPAA is a great scare word. Sharing de-identified molecular and clinical research data should not be a scary process. But we often now throw terms around like HIPAA and say, you can't do this, you can't do that. The right policies can counter data hoarding behaviors. Biologists, I used to be one myself, we don't love to share data. We'd love to get more papers on that data ourselves. There's a built-in kind of behavior that we don't like to share this. We don't need additional help in preventing us from sharing it, like HIPAA and others, okay? We need policies going the other way. But really the last point I want to make is we need new investigators who can ask basic questions using publicly available data. Why am I making a point of this? The first thing we train biologists to do sometimes is we tell them, don't trust other people's data, okay? That's usually the first thing we do to train a biologist. Generate the data yourself. And here what I'm saying is that we have all this publicly available data growing at an exponential rate, that we need a new generation of folks that are trained to trust that kind of data and know how to leverage. It's about leverage, right? Google only does well because everyone's on the internet now, right? That's leveraging off of that process. Same thing here. Everyone's using these molecular tools. Imagine the kinds of questions we can leverage into. And I want to spend a moment and talk about the role of the National Library of Medicine. It's probably the least understood institute in the National Institutes of Health. Yes, they give out grants. In fact, I got my start as a trainee under the National Library of Medicine. Now, they don't have a disease word in their title, like National Heart, Lung, and Blood, and diabetes, and cancer. But they fund grants. They launch investigators, but they're never remembered by everyone else who thinks about the disease side of it. I got my start with the National Library of Medicine, as have others. I just want to bring up the importance of that role of the National Library of Medicine. I talked about a lot of different pieces of work here, a lot of collaborators, specifically some collaborators in pediatric oncology and immunology at Stanford, close collaborators at the University of Tokyo doing genetics with us. I get all my funding from the Lucille Packard Foundation for Children's Health. I'm funded by National Library of Medicine now on an R1 at NIGMS, General Medical Sciences, and National Cancer Institute, as well as program project grants with NCI and NIAID, and pilot work with Genome Institute, Howard Hughes Medical Institute, corporate money from Hewlett Packard Pharma Foundation, that stem cell proposition we love in California. Yes, they even need bioinformatics, so we get some there, internal money. I always thank my administrative tech staff and my wife, who's a molecular biologist, who sees everything major comes out of my lab. I have to thank her in every talk as well. So thank you very much. Sure.
When you have the slides showing that heart disease and
muscular dystrophy, yeah.
Related, what sense were they related?
So specifically, when you look at samples with and without the heart attack, and with and without muscular dystrophy, the differences are pretty similar. So, in other words, the differences between heart attack and heart attack and the normals and muscular dystrophy normals are very identical. So, in other words, the process of getting that disease are so identical. Well, we give drugs to treat, sort of reverse the one. Why can't some of these drugs reverse the other? Is the question. At least it's a hypothesis. It's a question we can ask. Now, things are a little tricky in finding new uses for drugs because there's ownership and other issues there. There's a question over here.
Yeah, I'm still not quite understanding.
Surely.
Well, if you look at the tissue from a heart attack and the tissue from muscular dystrophy, are you simply saying they look the same?
That's right. I'm saying across the genome, they look the same.
Fine.
Exactly. Now, just to highlight that point, just to make it clear, heart attacks are treated by adult cardiologists. Muscular dystrophy is treated by pediatric neurologists, and these two kind of docs just never really even talk to each other. So they could be identical, and you wouldn't even know it today. That's the point here.
What kind of tissue would?
So one would be heart, one is muscle, from like a muscle biopsy. Question in the back.
This seems to be in drug companies and some features to find other uses for the drugs that they've already developed. How far along are they in going through this process? Can you give some kind of update there? Or whether it's not drug companies themselves, maybe researchers and those scientists.
So the question, I'll repeat it for the video too. So the question is, it should be in drug companies' interest, obviously, to find other uses for their drugs. And so if not even the drug companies, what about other investigators? It's actually common, if you actually study it, it's common for physicians who get an idea about a new use for a drug to actually put in investigation of new drug applications to FDA, actually get a use patent out of that. It's actually possible to do that, to get a patent filing and actually get approval to do that kind of study. You would think it is absolutely in the pharmaceutical company's interest to do this, but at the same time, I don't want to make this distinction between kind of them and us in academia. We are all subject to thinking about diseases in a very reductionist way. So, for example, when we think about cancer, we think about one type of cancer. Now we can think about 4 subtypes of cancer. And now we try to think about specific therapies for each of these. So I'm not going to fault the pharmaceutical industry at all, but we think about disease in a very reductionist way. We start with the disease, then we try to develop drugs against it. It is still rare for us to think the opposite direction. We have this disease. What other disease is it similar to? It's still rare to do that. I would absolutely agree. It is in the pharmaceutical company's interest to think this way. I think that's going to be a process both in academia and in industry to actually come up with new uses for these drugs. If you think about it this way, they're already approved for something. That means they're usually pretty safe in some ways too.
had a quick question for you. A lot of your work looked at genetic factors of disease. What about, say, for instance, epigenetic factors or, say, the emerging sort of role of proteomics? How do those...
That's great.
As
So
well?
the question is, a lot of people have been thinking about the genetic factors of disease. What about the epigenetic and thinking about other intracellular changes and things like that? Genetics for disease has gotten a kind of bad rap over the last 3 or 4 weeks. Multiple editorials in the New England Journal, as well as follow-up editorials in the New York Times, saying all these genome-wide studies haven't really found a lot of genes that are really implicatable for these complex disorders. The only point I want to make is this, that before we start to think about epigenetics and other kinds of genetics, let's not forget diseases are also caused by the environment, okay? Because we're just really often looking for molecular causes for these things, and they're environmental ones as well. Now, it's sometimes easier for us to say, oh, it's that gene that's causing you to have obesity. Okay, my friend Paul Wise on campus tells me this all the time. The minute we have a gene assigned to a disease, now we don't have to do anything about this disease, right? Because it's the genetics. That's why you're obese. That's why you're getting diabetes. It's not soda in the schools, lack of recess at schools, things like that. It's the gene that did this. And let's not forget the environment is there too. We don't have nifty little thumbnail-sized chips to study the environment. That makes it harder. But that's going to have a huger role in disease than the genetics today. Yes, there'll be a role. To directly answer the question, of course epigenetics will have a role. I think it's going to be trumped by the environment.
How helpful has the new public access policy at NIH been in your work?
Absolutely. So the new public access policy is incredible. In fact, from the investigator side, actually, it is a lot of bureaucracy to fill out, to upload your papers and things like that. But the NIH public access policy has really been about sharing papers and manuscripts, especially upon acceptance, to get your raw manuscript out there so everyone else can read it as well. What I'm asking you to think about and consider is an additional level of detail now, not just sharing the papers, but actually sharing the raw data. And that the public access policy actually doesn't cover today. It's more the journals and certain funding agencies. For example, if you apply for an NIH grant more than $500,000 a year, you have to put a data sharing section. Nobody even knows how to review those. Nobody even really makes you follow up on what you said you would do. But so there's some thought there. It's mostly the journals right now that are asking the data to be shared. And that's not really the most efficient way to do this. We're probably still only capturing maybe 1 to 10% of the actual data that's collected. I just think there's a world of amazing findings that are left out there that we really could use to create these diagnostic therapeutics if we were more efficient about sharing the data.
So as these databases grow and you are able to form these virtual cohorts and do virtual clinical trials, essentially, does informed consent have to change? I mean, so it's already difficult to communicate with participants what exactly they're consenting to, and now their data is not going to be used for a diabetes study. Only it might be used in some other study. Do you have to go back and reconsent?
That's right. That's a good question. So the specific question is, what about the informed consent aspect of it? Especially when data is collected for one particular study that they might have really accepted and even were enthusiastic about is now used for a different study. In some ways, so there's going to be a lot of these types of issues that have to be sorted out. But in the majority of cases here, this is de-identified data. Okay, so to be honest with you, most of the research I'm talking about here is actually not even considered human subjects research because it's public data anyone can get access to, and it is impossible to re-identify the patient from that. So there are these there are a lot of technicalities when you look about how to get samples and data from patients in terms of discarded samples and things like that. These are de-identified data where it's not, so it's publicly available in that kind of way.
So as a follow-up to that, the de-identified data, though, doesn't inform the environmental aspect of your research. So how do you then get at, if environmental is the bigger part of this, how do you study that if you can't link data to a geographic area or data to a specific ethnicity or data to a community? Yeah, so so how do we get the environmental aspects of it if it's fully de-identified? There's still other ways you can imagine to de-identify other pieces of the medical record at the time when the sample was collected. I think the real hard problem is when you have a sample and you're getting more data on that patient and now you want to use that new data on a patient. There's still ways to share, there's still ways to de-identify, there's all sorts of creative ways to de-identify exactly where a patient lives, what zip code they're in, census tract, not to mention how the distances that they live from other various point sources of pollution, things like that.
what school do they go to, what's their body mass index. Those are all numbers in some ways that can easily be de-identified. What I'm saying, though, is it is not impossible to do that. But I think we throw up HIPAA and say, well, let's not even bother trying sometimes. And that's where I think we're actually at a disadvantage, unnecessarily.
With President Obama's focus on electronic medical records and moving towards making more data available for improvement of human health, how much do you think people like yourself can be involved in the conversation about how to make data available for research purposes?
Yeah. So with President Obama's push towards making, with really having electronic health records for all individuals, all Americans, not to mention for research purposes and things, how can the discussion be had? I think there are a lot of discussions being had. So that is kind of a problem in itself, where there's various different organizations in terms of harmonizing with clinical trials. There's the clinical researchers, pharmaceutical trial folks, clinical research informatics folks, a lot of folks having these discussions on how to do this. It does definitely help to have the mandate that this should be done, right? So it's funny, in some ways, it is amazingly easier to share genetics and genomics data than the clinical records, right? We still don't have any public repository, let's just say, of 100 electronic medical records, completely de-identified. Even 10 medical records. It's a little strange when you think about it, why we can't even start with small numbers here. I think what happens with us in informatics and computer science and engineers is we try to over-engineer the perfect solution. Well, the way I call this should be the same way that you call this. And I'm for actually more simpler, faster solutions. So get some use cases, let's get some real tractable findings out of this, and then we build from there. The same way the internet kind of grew. We didn't have a committee to make HTTP or anything. It was one individual who did this in the end, or a handful of individuals. So I think we can sometimes be guilty of over-engineering systems, but I see the discussion is absolutely happening now.
Isn't the NIH's eMERGE project, you already bit to what you said?
Yeah,
and,
that's great.
And they struggle with defining the phenotypes.
Absolutely. So the NIH eMERGE is a great point. So eMERGE is a coalition, I think, between, certainly Vanderbilt is the major lead, Marshfield Clinic in Wisconsin, as well as Northwestern as well.
University of Washington.
Yeah, so this was a coalition of research institutes. Yeah, there's one or two others.
There's another one that I forgot.
They're going to kill me now. So it's a coalition of institutions that had biobanks set up. So they were already collecting DNA, if not other types of tissue, prospectively on patients, and medical records, and de-identifying them. Now, first of all, it's sort of embarrassing there's only a handful. Mayo Clinic, of course. It's embarrassing there's only a handful of these institutions that actually have thought of doing this, prospective collection of samples, but it's a hard process. You can imagine to de-identify an entire electronic medical record, capturing the phenotypes correctly, and then connecting them to the actual samples, getting rid of duplicates without knowing who the patients are. It's a hard process, but it is being done. Now, eMERGE is great, but again, it's great for eMERGE researchers in the end. They will certainly be able to share data within their own community. But at the same time, how that's going to scale to others is an open question still today. At Stanford, for example, I cannot get access to eMERGE data. We're not as forward-thinking, I guess, with the biobank.
I have a question. I apologize if you talked about this in your talk, but
surely.
About this, organizing things. Now that you've found this link between muscular dystrophy and heart disease, what are the next steps, and are next steps being taken? I mean, is this the work you're doing really being translated to some level? Yeah,
so certainly the easiest way to think about joining diseases is certainly on the diagnostic side. So that's an example where we take diseases, heart transplant doc and kidney transplant doc should talk to each other. There's these common markers. The diagnostic side is easier. The clinical trial side for the therapeutics is obviously a lot harder. We're trying some of these. We're launching some mouse models we actually have some predictions of over-the-counter medications that we think actually should be treatable for lung cancer. We're going to try those out and see if we can do that. And then eventually take them either in private or academic partnerships to see if we can get some of these trials done. Now, in theory, it should be smoother because they're already approved. They've already gone through a phase 1 study. It's just a matter of figuring out a new phase 2 study for them. But I think so far, no roadblocks. It's obviously going to take longer than the diagnostics on that side.
I'm going to rudely call out Congressman Rush Holt, who's been sitting here quietly, and I don't know if he's even prepared to talk.
Sitting here interested. Thank you for an interesting presentation. And just to remind the audience here, the reason we have these biomedical lunches, they're interesting, intellectually stimulating, but of course that's not the point. The point is to draw attention to the support that NIH and other funding agencies have provided that make this possible. I don't know that you're prepared to say anything more about the Library of Medicine,
absolutely.
What its funding status is, and if you wanted to talk about its financial health, relatively speaking.
Yeah.
I think people here would be interested.
Great.
But it's, we often don't think about how the benefits to society come about. The NSF grant in library science for databases that went to a couple of guys, 15 years ago or whatever it was, resulted in Google. It was I'm sure none of the reviewers thought that it was going that way. But it was well, for one thing, it shows the power of library science, which is, in a sense, what we're talking about here. But it also shows the importance of having federally funded research that is not overly directed.
That's right.
And and NIH has been very good in many cases in that respect. Well, maybe you want to say something about the library.
Sure. So these are going to be numbers off the top of my head, but they're easy numbers to remember. So the National Library of Medicine is an IC, sort of an institute center at NIH, of the 28 or so of them. But I don't need a list to show me. I can guarantee you their numbers are at the bottom of the list. But they actually have the impact of any of the other top-tier institutes. So they have a K Award or Career Development Award program, the Pathways to Independence program that they fund. They fund R01s, which are standard program project grants, plus larger multi-center grants. But they're doing this on in ridiculously tiny budget. I'll be honest with you. I don't have the numbers in front of me, but it's got to be a tenth of NCI, NIGMS, and the others. And plus, what's worse, in my view, is that when increases are called for, which is great for NIH, they seem to be forgotten all the time. So there's they're just not thought of as another institute.
Every one of your offices here has had, in the last week, somebody coming through asking for increase in funding in a particular disease.
Right.
I have not, in my 10 years in Congress, going on 11,
had a librarian.
Had anybody come
That's the problem.
that I can think of to advocate for funding for the Library of Medicine.
That's why it's the lousiest name you can imagine. To have a library, you all have memories of libraries and what they're like. This is not like that. So the NCBI, the National Center for Biotechnology Information, is the hotbed. We have PubMed, we have the GenBank, we have all of these repositories. That's not even intramural. That is a work product of the government. That's their daily job at the NLM, is to build those repositories. Plus, they have intramural research and extramural research. I'll be blunt. I would have not gotten my start in research had it not been for a Career Development Award from the National Library of Medicine. But people forget about National Library of Medicine all the time. It is just like the NCI and NHLBI. The budget's a lot smaller, and its fraction is diminishing because it doesn't benefit from the increases of others. So I don't know if there's other thoughts, if anyone else has questions about the National Library of Medicine. I'm certainly not the best representative, but I think I wouldn't have had my start if it weren't for that institute. Other questions? Great.
Thank you.
Thank you very much. Thanks for coming.